Hi everyone, thank you for joining us today. Another installment of the Illumina Webinar series. For those of you that this is your first time, you can ask questions in the lower right hand corner. Just type your question in there. If you would like to see the slides in a larger slide mode, you can hit the expand button in the upper right hand corner next, just above the slides. You'll lose the ability to type in a question, but you can just hover over that area, minimize it, and then you'll be able to type any questions that you would like to ask at the end of the session. So today we have Dr. Lachlan Jolly. Dr. Lachlan Jolly completed his PhD at the University of Adelaide in South Australia in 2010, where he applied neural differentiation of embryonic stem cells to study the earliest stages of brain development. From there, he joined the neurogenetics research program headed by Professor Joseph Getz for postdoctoral training at the Women's and Children's Hospital in Adelaide. His application of various neural cell culture models of brain development has resulted in the discovery of several new genetic causes of neurodevelopmental disorders. Dr. Jolly now leads his own research group at the University of Adelaide, focusing on the roles of nonsense-mediated mRNA decay, or the NMD pathway, and the genes USP9X and HCFC1, and their roles in normal and pathological processes that affect both brain development and function. He is currently an Australian Research Council Fellow, and we're really happy to have him here today. The title of his talk is Investigating the Effects of Genetic Change Using Cell Models of Brain Development. Dr. Jolly, welcome, and the floor is yours.
Thank you very much, and I'm really happy to be giving this seminar today. Thank you for those who have logged in to be part of this. So I'll move straight on into, I guess, a bit of a general background. The main mantra of our lab is to improve the quality of life for people suffering with neurodevelopmental disorders, and my particular focus has been individuals with intellectual disability and epilepsy, and predominantly been focusing on those families which show linkage to the X chromosome. The process begins for us to really, when we have families that have this inheritance pattern where we have affected boys and females that are generally unaffected, and then to really go into the genome and using massively parallel sequencing to try and find genetic variants that may be the root of these disorders. Of course, this information, when we receive it, is really handy to give back to the family that can help them with counselling for family planning and stuff. But often the genetics is perhaps not as convincing, so diagnosis is left, where we really need to then have a look at variants of unknown function. So these are variants which are perhaps pathological, but perhaps not. One way that we can try and gain some more information is to really see whether these variants in genes actually affect the ability of these genes to function. And the model systems I like to use are those that actually model development of the brain. Of course, this information, when we get it, is really helpful as well, because without this functional data and understanding how these genes really do play a role in brain development, is the only way in which we can begin to come up with potential therapeutic targets and regimes that can actually feed back and help the affected individuals of the family. So for me, the focus of my research is really understanding the function of genes involved with these disorders and in the normal and abnormal development of the brain. So the materials, I guess, available to me to complete these studies are varied. We have cell lines that we can get from affected individuals, whether these be blood or skin derived, and these, as I mentioned, are a really good source of DNA for us to begin the gene discovery, but perhaps not so conducive to really studying developmental processes and cell and molecular biology. For that, often if we really want to know the function of a gene in in brain development, we can turn to small animal models, mice, zebrafish, and etc. And these are fantastic for studying developmental process, but they're very hard to manipulate, and there's quite a large bit of investment involved in setting up these systems. So another way in which many people use is to use cell culture models, and there are a few cell lines out there which do indeed present some recapitulation of developmental events, whether it be a neural stem cell line that can differentiate into a neuron or into a glia cell, and these cell lines at least give us some information on developmental processes. Of course, more recently we have these cell models, or these resources, I guess, are interconnected, and in particular induced pluripotent stem cells, or even CRISPR editing of human embryonic stem cells gives us now the power to take actual human-derived tissue from affected individuals and then use differentiation of these cell lines into various neuronal lineages to study. Again, this has a lot of power, but also it's also associated with a lot of costs. Another system is to actually take cells out of the developing mouse brain and culture them in vivo. And this is something which I'll touch on throughout my talk. I find this system a very useful way to study in a quite quick and economical way. I guess once the cells are taken out of the mouse, they still have the developmental potential. They still differentiate and undergo processes in the dish that recapitulate brain development. And because you've taken them out of the mouse, they're very easy to manipulate, which means that we can assess the effects of genetic change quite easily. So getting to, I guess, back to where this talk sort of begins, in 2009, I was involved in a publication which looked at a number of, trying to find the genetic cause of intellectual disability in a number of families that displayed X linkage. So here you can see the square boxes are the males, and they're blacks because they're suffering from intellectual disability, and you can see throughout these families that the females in circles are carriers and are unaffected. So this really told us that the X chromosome is likely implicated. At this point in time, massively parallel sequencing had not really become accessible. And so we were part of a cohort that actually used Sanger sequencing to sequence the whole X chromosome coding regions to try and search for genes in these families. So this had a number of outcomes. Of the 208 families which we studied, we found mutations in this gene, UPF3B, in a number of families, which is quite compelling evidence that this gene and the mutations in this gene, which were mainly loss of function, and in some instances complete knockouts, was involved. For other genes like USP9X, there was some indication that the gene could be involved, but not a lot of other genetic evidence to suggest it definitely was. And for some families, like this one on the right, despite sequencing the whole coding region, we still did not have an answer of what gene was implicated. So today I wanted to talk to you about two of these genes, the UPF3B and this undiagnosed family on the right. And so in a way, the talk is split up into two parts. Okay, so for UPF3B, now this gene is a member of the nonsense-mediated mRNA decay pathway. So I just thought I'd put up a few slides about this cellular mechanism, which is really important for the degradation of transcripts that harbour premature termination codons. So UPF3B, you can see here, is part of the exon junction complex, which is deposited on all mRNAs post-splicing. When this mRNA is transported out of the nucleus, it's joined by a second core NMD factor known as UPF2. Now, in normal transcripts, during the pioneer round of translation, the ribosome scans along, and when it runs into these exon junction complexes, it displaces them. And because the stop codon in the majority of all eukaryotic genes is encoded in the last exon, by the time the ribosome hits the stop codon, all of these exon junction complexes have been removed. Now, it's this fact that all have been removed when the ribosome stops, that has been sort of leveraged in a way by the NMD system to identify transcripts that have a premature termination codon. Because here you have the unusual scenario where you have a stalled ribosome and an exon junction complex downstream. It's this unusual architecture that results in the recruitment of a third NMD factor, UPF1, and which basically coordinates numerous protein complexes that result in degradation of the mRNA. So in a way, this system was first discovered for its ability to degrade transcripts with premature termination codons, in a way protecting the cell from the potentially deleterious effects of a truncated protein. And because of this, this pathway is actually implicated in thousands, in fact 1/3 of all genetic disorders, which are actually caused by premature termination codons. But it turns out that this pathway, whilst it has this surveillance process, is actually more important. And it turns out that there is a large number of endogenous transcripts that have features in them that are consistent with regulation by the NMD pathway. For example, there are transcripts that have upstream open reading frames. And again, this provides a scenario where we have a stop codon upstream of a exon junction complex. Termination codons can be introduced by alternative splicing events, and some transcripts actually have introns in the 3' UTR, and in this case, the actual stop codon, again, because it's upstream of an exon junction complex, can be interpreted and targeted by the NMD pathway. So, how many transcripts are like this? Well, in fact, if you knock down UPF1 or UPF2 in cultured cell lines, you end up getting around about 5% of the transcriptome deregulated. So that's a very large proportion of genes which are directly or indirectly affected by this NMD pathway. And these genes are actually important for development, because if you knock out UPF1 and UPF2 in mice, in zebrafish, in Drosophila, you actually get an embryonic lethal phenotype. And as I mentioned at the top, we became interested in this pathway because we found that mutations in UPF3B, and some of these are complete human knockouts, results in intellectual disability in man. When we further looked into this pathway to see whether the other components of the pathway were involved also in intellectual disability, what we found was numerous copy number variants, either whole gene deletions or copy number gains of various components of the exon junction complex, the NMD core machinery, that were actually enriched in neurodevelopmental disorders. So whilst we began with UPF3B as our main interest, we're now of the opinion that the whole NMD pathway could be very important for normal brain development. So how do we begin to work out what's the sort of underlying pathological mechanism? Well, we took advantage of our cell lines that we had actually derived from our affected individuals. You can see 4 of them here. We collected blood cell lines from them, and compared to 2 normal healthy control individuals, we compared the transcriptome using both RNA sequencing and also microarray. We also screened the DNA for any other copy number variants that could somehow explain changes in this gene expression, and really then looked to see which particular genes, or what was the degree of alteration in the transcriptome, and also which particular genes might be deregulated in blood that could give us some sort of surrogate information onto what was happening in the brain. When we looked at UPF3B mutations, we found, again, 5% of the transcriptome was deregulated. So this was consistent with the previous studies which I mentioned. And in this instance, 60% of the genes were upregulated, which is what you would hope to expect given that this process degrades transcripts. But we also have 40% of them which are downregulated, so there are some indirect targets or downstream effects. As I mentioned, this was for the patient, well, for the individuals that had alterations in UPF3B, but we also had individuals that had copy number loss in UPF2, and we wondered whether there was a shared signature in transcriptome changes between these two different groups of individuals that both had deficiencies in the NMD pathway. So we can see when we compared the deregulated genes, there was a reasonably large overlap of 379 genes that were significantly deregulated by twofold that were shared between both of these groups of people. But perhaps more convincing, if we look at the figure on the right, is that the trend of deregulation in both these groups was the same. So in UPF3B affected individuals, where you have downregulation, we also saw those same genes downregulated in the individuals with UPF2 copy loss, and the same with upregulated genes. So the correlation of changes in gene expression was very similar between these two groups, again suggesting that various insults to this NMD pathway have a shared and common underlying deregulation that leads to a common way in which the brain development is abnormally affected. So despite being from blood origin, there are actually genes that we picked out that were significantly deregulated that had bona fide roles in the development of the brain. So we can see various genes involved in nervous system development, and a couple of other ones which we were really interested in. This one here, RGAP24 down the bottom, we actually show that if you manipulate the expression of this gene in neurons, it really had quite a toxic effect on the neurons and changed their morphology quite drastically. So that kind of suggested to us that, you know, despite being a blood origin, that we were looking at genes that were involved with brain development, but we really wanted to perhaps take a more direct approach and really ask what is the effect, or what is the main role of UPF3 dependent NMD during brain development. So we really wanted to know exactly where this pathway was most active, what stage of brain development is this pathway really important for, and once identifying that stage, what would be the effect of reducing UPF3B in these cells and seeing if we can find some sort of readout that could help us understand the pathology in the affected individuals. So where to look? What we decided is that, well, we wanted to know where UPF3B dependent NMD was most active, but before that we were aware of some information. Well, it's not just the expression of UPF3B that we were after, because we know there are modifiers to the outcome of nonsense mediated mRNA decay. That being that UPF3B has a paralog called UPF3A, and they are somewhat functionally redundant, so that in the absence of UPF3B, nonsense mediated mRNA decay can still function at a certain level because UPF3A can sort of functionally compensate for this loss. The other pathway which we were interested in is also somewhat of an indirect inhibitor of the NMD pathway, and this is something called Stauffen-mediated mRNA decay. So Stauffen can bind a separate set of transcripts through a cognate binding sequence in the 3' UTR, and the Stauffen-1 actually in a way competes for the NMD machinery. So there's a stoichiometric sort of competition for UPF1 by UPF2 and Stauffen-1. So in a way, in cells where the SMD pathway is really active, the indirect effect is that the NMD pathway is somewhat suppressed because it just cannot get access to UPF1 and the shared degradation machinery. So whilst we wanted to have a look at UPF3B expression, we also were aware that the output of the NMD pathway would be a function not only of UPF3B expression, but also of Stauffen-1 and UPF3A. And so when we stained the developing brain with these genes, with antibodies towards these proteins, we saw definitely that there was evidence that these different pathways were acting perhaps in a dominant or sort of passenger way in the cells. For example, I'll just take so down here, this is developing forebrain at E14.5. where I've got the pointer here is the neural progenitor cells of the developing brain, and you can see they're highly enriched with the Stauffin-1 protein, whereas the cells out here, which is where the newborn neurons settle in the developing brain, are really highly enriched for UPF3B. Likewise, when we go a little bit further to E18.5, down here now is the ventricular zone. It's much smaller, but you can now see that the Stauffin-1 protein is beginning to reduce and UPF3B protein beginning to be enriched. And you can see throughout this cortex that there are different layers of cells which have different dominant expression. So here are some cells that have UPF3B and UPF3A. Out here is another layer which seems to be more strongly expressing UPF3B, and out here again, Stauffin-1 seems to be dominating. So there's certainly an interplay between these different pathways and what is dominating, and it seems to be stratifying the cortex in a way that might be demarcating particular neural cell types. I guess more specifically, when we're looking directly just at UPF3B, we also noticed that there was some changes in protein localization. So at E10.5, this is all neural progenitors. You can see the localization is predominantly cytoplasmic, which again seen in the ventricular zone here at E14.5. But when we get to E18.5, which was, as I mentioned before, correlated with an increase in expression in the ventricular zone, we also see a switch to a more nuclear localization of UPF3B. So the expression and localization of this protein is quite dynamic. And I guess the take-home message for us was that we correlated increased UPF3B expression with ongoing development of the forebrain, which is associated with decreased proliferative capacity. And in fact, out here at E18.5, we had both high expression and we had also nuclear localization, which we perceive as a prerequisite for UPF3B function. And so what we really decided to do was to take these cells, the progenitor cells from the E18.5 embryo, and conduct loss of function experiments in the dish to see what would be the effect of loss of UPF3B. So we took wild-type mice. You can take the progenitor cells out and culture them as non-adherent neurosphere cultures, where essentially the cells grow as a floating ball of cells. You can dissociate these cells and reculture them. And what happens is they reform these spheres over time. But not all cells form the spheres. It's only this rare population. Around about 5% of the cells, this green cell here, have the properties of a stem cell. And it's only these cells with actual stem cell long-term self-renewal and multipotency that are able to reform spheres. So we took these neurosphere cultures, we infected them with lentivirus encoding either control or UPF3B specific short hairpin RNA, and we collected these cells by fax because there was a fluorescent marker. And with these, now we have these neural progenitor cells in the dish lacking UPF3B function. So one of the first things we did was to have a look at the biochemistry. We collected the cells, and this is just, I guess, evidence that we were knocking down this UPF3B protein. In all the experiments, we have 3 control conditions and 3 UPF3B knockdown conditions using different triggers. So UPF3B is knocked down, and on the right here, you can see that when we look at some of the bona fide NMD target genes, that they are actually upregulated in these cells also, suggesting that not only have we knocked down UPF3B, that we've actually compromised the NMD pathway in these cells. So having these cells gave us an ability to quickly look at those alternative and competitive pathways, which I mentioned previously. So we can see that UPF3A expression was upregulated in these cells, suggesting that in the absence of UPF3B, this pathway can sort of compensate. We saw that there was increased expression of Stauffin-1 at the mRNA level, and we looked for evidence of increased Stauffin-mediated mRNA decay, and at least a couple of these genes, SERPIN1 and GAP43, which are SND targets, seem to be downregulated, so perhaps suggesting that SND was in fact increased in the absence of UPF3B. So as I mentioned, the data here certainly suggests that one of the normal functions of UPF3B is to suppress the alternative NMD pathway that utilises UPF3A, and perhaps also in these particular cell types maybe to also inhibit SALFN1 mediated mRNA decay. In fact, if I just go back, this interplay between UPF3A and UPF3B we think is really important for the presentation of the pathology in patients with UPF3B mutations. So here are some affected individuals with loss of function UPF3B mutations. You can see P1, 2, 3 and 4 are all affected individuals and they do not have UPF3B protein. But you can see normally where UPF3A is absent in these cells, and these are patient-derived cells, in the absence of UPF3B, you can begin to see that UPF3A becomes stabilised. And in fact, if you have a look at these two affected individuals that come from the same family, one has quite low UPF3A, and they have a quite severe phenotype, whereas the other brother has quite high levels of UPF3A, and their phenotype is quite mild. So it's like UPF3A can rescue the severity of the disorder. And we're actually able to show this at more quantitative level, looking at the level of mRNA degradation. So here is the control. This is just looking at the average expression of all genes that were upregulated. And you can see here the brother that had low UPF3A, a more severe phenotype, his deregulation is far more different than the brother that had high UPF3A and a less severe phenotype. And we also see the same trend for the downregulated genes. So UPF3A seems to be a modifier of the disease outcome. So that was interesting. Of course, we wanted to look at how the loss of UPF3B affected the behaviour of these cells. So we went back to our cell culture assay, and these neural progenitor cells, you can do a couple of different things. You can ask what's the effect on the ability of these cells to keep self-renewing in the neurosphere culture condition, and you can also ask what is the effect of their ability to properly differentiate. So when you plate these cells out onto a substrate and remove growth factor, they differentiate into neurons, astrocytes, and oligodendrocytes. And you can monitor kinetics of this process. So looking at how the neurosphere cultures went, we did just a simple cell proliferation assay, and when we knocked down UPF3B, we can see we had an increase in cell numbers in these cultures. So we really wanted to dig down to work out what could be the explanation. We looked at whether there was an increase in the percentage of proliferating cells. We looked at cell cycle. We looked at apoptotic rates. I can say the last thing that we looked at was the ability, well, the number of stem cells in these cultures, and that actually gives us an explanation. So just to show you that data, as I mentioned before, only a really rare population of cells in these neurospheres can actually reform a sphere. And so when you plate out a set number of cells, say 1 by 1 million cells, and then you ask how many spheres are formed, the number of spheres that form is actually a direct readout of the number of stem cells that you plated out. And so if you count spheres over serial passages, we can always see that when we knock down UPF3B, we always got more spheres forming in the cultures over several passages, and this was highly significant on average. So this, to us, pointed out that in the absence of UPF3B, that we're actually increasing the number of neural stem cells in the cultures. So, as I mentioned, we had a look at self-renewal, but the other thing we can do was to have a look at if we have increased self-renewal, then perhaps we also had a decrease in the ability of these cells to differentiate. And for that, as I mentioned, we plate the cells out, we let them differentiate over a few days, and then we can use marker genes to stain the cultures. PAX6 stains for neural progenitor cells, beta-3 tubulin stands for early born neurons, and GFAP stands for the astrocytes, and really just conduct cell counts to see where there was a difference in the kinetics or composition of these cultures. And we can see that when we reduce the expression of UPF3B in these cells, that there was, again, an increase in the number of progenitors at the expense of the number of differentiated cells. So we really wanted to, I guess, reconnect our in vitro models of this disorder back to the patients or back to the affected individuals. And we did this by having a look at some of the deregulated genes which I mentioned before. So these are a list of genes which we discovered in the affected individuals' blood cells that looked like they had roles in brain development. So we took some of these genes. The arrows on the side, on the right, indicate whether they had an NMD feature or whether they did not. And we took some of these genes with the stars next to them and analysed their expression in our cultures. And what we saw was one particular gene, 6-3, was upregulated 3.5 times in the absence of UPF3B in these neural progenitor cells. And this was quite interesting to us because when we looked back at the literature, 6-3 expression, when you overexpress it in neural progenitor cells in a very similar neurosphere system as we did before, you get exactly the same phenotype of increased proliferation at the expense of differentiation. So that led us to somewhat of a working model that really, as cells of the developing brain lose their proliferative capacity, that you get an increase in the increase of the activity of the UPF3B dependent NMD pathway. And we think that this association or correlation is actually functional because when we knock down UPF3B expression, we endow the cells with a more proliferative capacity. We also think that part of this mechanism involves 6-3. Of course, there'll be many other genes which the NMD pathway will target, so this is not the whole story, but we think together the cassette of genes that the NMD pathway is controlling is really shaping the transcriptome of these cells in a way that's more conducive to the differentiated state. I guess just to go back to the bigger picture and to finish this half of my talk off, is that we really began the story knowing that UPF3B alterations caused a range of disorders, predominantly all featuring intellectual disability, but also ADHD, autism and schizophrenia in affected individuals. And we really wanted to try and fill in the gaps between knowing a mutation and knowing a clinical phenotype. So we looked for what would possibly be modifying, we looked for the NMD targets and what cells and processes were affected. We were beginning to fill in some of this information. For example, UPF3A we really think is a potential modifier of the disease outcome. I showed you a mechanism today involving 6-3 and the progenitor cell self-renewal that could perhaps explain features of the phenotype, which is known to include macrocephaly. But there are other data which I haven't showed you that we also did similar studies in neurons and showed defective growth, and there are also target genes which can explain this. So we're really now looking at expanding this quite focused study into actually moving it into a more humanized system, and that being editing, knocking out these genes in human embryonic stem cells and really seeing if we can target in on perhaps a larger overlap with the actual human patients by using human material to begin with. So this is using human differentiation of human embryonic stem cells into human neurons to study. So that's going forward. I'll bring you back to this slide. As I mentioned at the top, I wanted to talk about two quick stories today. The second story was this very large X-linked family on the right here, and despite us sequencing the whole exome, we were really unable to find a gene or a variant in any gene that could explain the phenotype of intellectual disability. So this family was even bigger. This is the whole pedigree. It was in fact first described in 1991, and because the pedigree is so large, and you have members of the family that are very separated by many generations, you're able to do linkage analysis and really focus in on part of the chromosome that's likely responsible, because this part of the chromosome is shared between all affected individuals. So now, instead of looking at the whole X chromosome, we're really focused in on this XQ28 region. So we thought what we would do is really focus in on this region. Here it is blown up. It contains 108 genes, which are listed here. This is just the UCSC tracks. And what we decided to do was design a tiling array where we could capture these sequences for all of the coding and non-coding regulatory regions around these genes. So that enables us to enrich the DNA many-fold over, and so that when we're sequencing, we're only really sequencing the part that we've tiled. So we're really focusing in, getting incredibly deep coverage, and that enabled us to really, I guess in a way, double check and also check into non-coding regions if there are any variants that could explain the disorder. Well, I can tell you, after we were a little bit disappointed at first because, again, we came up empty in the coding region, so there were no coding variants. What we did come up with, though, when we looked just outside of a gene called HCFC1, was a very well conserved change in a non-coding region. So this change, this simple G to A change, actually affected the binding site, well, it changed the cognate sequence of a binding site of the YY1 transcription factor. And when we had a look in patient cell lines, indeed what we saw was that the expression of this gene HCFC1 was upregulated in the patient or in the affected individual's cell lines. We went on to show that YY1 indeed did bind this site. We used electromobility shift assays, and we also used chip assays to show that YY1 actually bound this site, and if we introduce this variant, then that abolished the ability of YY1 to bind there. Furthermore, we used luciferase assays to really show that YY1 in fact did repress HCFC1, and that if we introduced this variant into the luciferase reporter, that the luciferase expression went up. So the model is that HCFC1 indeed binds this site, and its normal role is to repress HCFC1. But with the variants in place, this repression is lost, and we get overexpression of HCFC1. So HCFC1 itself is a transcriptional coactivator. It essentially acts as a bridge between transcription factors and histone and chromatin modifying enzymes. So here it's best characterized for its role in the cell cycle, and it can have both positive and negative roles on transcription. The gene itself has many different domains. It's actually cleaved in the center and acts as a homodimer of NMC fragments. So because it is a transcription factor, again, we turned back to our cell lines that we derived from affected individuals to see whether we could reveal any transcriptome changes and whether these transcriptome changes would be, I guess, in a way, reflective of an alteration in HCFC1 function. I must say, I mean, we found the regulatory mutation, that's okay, but we were by no way convinced that this change was the actual causative, the causative variant. So we took, again, lymphoblastoid cell lines from affected individuals compared to controls. In this instance, we used microarray, and we pulled out 218 genes that were significantly deregulated. What was pleasing to us is when we did gene ontology analysis of these genes, that we were pulling out pathways that HCFC1 was known to regulate, that being chromatin modification, also the control of the mitochondrial function, and somewhat to our surprise, but what pleased us, it also highlighted unknown functions for HCFC1, and that was a number of genes, again, coming up with the gene ontology pathway that was highly enriched was forebrain development and neuron development, which were really, again, pointing to us to to suggest that perhaps using the neurosphere or ex vivo cultures of neural development might actually be a way to assess the function of HCFC1 to really show that it had a role in brain development. So I've been through the neurosphere culture before, just previously, but we essentially applied a very similar regime. But in this instance, we also, based on expression, knew that HCFC1 was highly expressed in the developing hippocampus. So another assay that we ran was to take the neurons, instead of taking progenitor cells out, we go in and actually take the neurons from the hippocampus, and when you plate them out, they follow a very stereotypical developmental process in the dish where they regrow their neurites, they specify an axon, and then they really elaborate. And in fact, over the period of 2 weeks or 3 weeks, they actually form synapses and begin to form a functional network. So this kinetics of axon growth and neurite growth you can study in the dish over the first week or two of hippocampal cultures. And of course, we were wondering what would be the effect of overexpression of HCFC1 in this instance. So I guess just briefly, we saw that when we overexpressed HCFC1, we forced the cells to exit the cell cycle and become differentiated. It's perhaps best shown on this bottom here. You can see cells which are overexpressing GFP, the cells which are transfected with HCFC1. And if we have a look at the co-localization of these transfected cells with the astrocyte marker GFAP, we can see that in controls, they don't really overlap. But when we overexpress HCFC1, a large number of astrocytes are expressing GFP. And so essentially, by overexpressing HCFC1, we're again changing the behavior of these cells and forcing them to differentiate. Likewise, for the axon growth, you can just have a look at the kinetics of axon growth here, that when we overexpress HCFC1, we actually inhibit the ability of the axon to grow properly. So that for us was fairly solid evidence, both functional and some genetic evidence that HCFC1 was involved, likely the solution for that family, which ended over a 25-year diagnostic odyssey for them. But we're always keen to find more evidence, and more evidence came when we actually became aware and discovered more variants in HCFC1 that also had intellectual disability. But in these instances, these variants were actually in the coding regions, so these are missense variants. So slightly different to the non-coding variant that we studied before. And in fact, one of these variants affected the nuclear localization sequence. So it suggested in a way that these variants perhaps also include some that have loss of function. One of the first things we did was to clone these variants into an expression vector and just see where they localized, with particular interest in the nuclear localization variant. So here, the schematic at the top shows that the expression contract we used had a HA tag and a MIC tag at both ends of the protein, and as I mentioned before, this protein is cleaved and can act as a dimer. We also have an antibody raised to the endogenous region, just here in the C-terminal spot. And so here is just what endogenous HCFC1 looks like by immunofluorescence. It's in the nucleus. When we overexpress wild-type HCFC1, we can see that the MIC and the HA fluorescence overlaps in the nucleus, again overlapping with the endogenous expression. And when we overexpress the variants, we essentially saw exactly the same features, except for the nuclear localization variant. You can see quite clearly here that both the N-terminal tag and C-terminal tag are predominantly, not completely, but predominantly excluded from the nucleus. So certainly, this was telling us that we have a different sort of variant here. These, as opposed to our initial discovery of a non-coding variant which caused overexpression, these particular variants look like they were loss of function. Well, when we were preparing this work, I guess we were paid particular interest to this article from Tamim Shakik's lab in the University of Colorado. Now, he discovered mutations in HCFC1 in a slightly different region of the protein to us that caused a metabolic disorder called common lamina disorder, which is essentially a vitamin B12 deficiency disorder. Most commonly, this disorder is caused by mutations in this gene, MACHC. So they decided that it would be interesting to try and link HCFC1 function to MACHC, and they didn't do this. So on the bottom here we can see some graphs. The first two bars are the affected individuals with the common lamina deficiency, and the second two bars are the controls. And you can see in the HCFC1 patient cell lines that MACHC expression is essentially lost. So that was a nice link for them to their common lamina deficiency. Now, common lamina deficiency, what they did also, I should mention, is showed that their mutations were loss of function, because you get the same loss of MACHC expression when you knock down HCFC1 function. So typically with this vitamin B12 deficiency, you have quite mild neurological features. But in these particular patients that had the common lamina deficiency as a result of HCFC1 loss of function mutations, the neurological features were very severe, including intractable, like epilepsy that couldn't be treated, and very severe intellectual disability. Of course, now we were wondering, certainly it looks like loss of function mutations in HCFC1 can cause neurological problems. So we really wanted to go back to our original assays that we set up to have a look at HCFC1 function and ask what was the effect of knocking down HCFC1. And the short story is that when we knock down HCFC1 expression, we see kind of the opposite effect. We see increased progenitor proliferation and increased axon growth. And in fact, this increase in axon growth gave us a nice assay to test the significance or the functionality of the other variants that we had discovered. So we took neurons, we nucleoaffected them with either control, wild type, or one of the 4 variants that we had discovered. And we also then co-transfected in or transduced in a lentivirus that could either knock down or that could knock down HCFC1. And what we saw when we did this assay was that 3 of the 4 variants essentially behaved as a loss of function. So this is just similar data to before, that when we overexpress HCFC1 here, we get a reduction in axon growth. And essentially when we overexpress 3 of these 4 variants, this suppression of axon growth is missing, suggesting that they are loss of function. And we get exactly the same data essentially when we use the short hairpin. So when we knock down HCFC1, we get an increase in axon growth compared to control, which we could rescue when we overexpress wild type and this valine variant, but the other 3 variants that we tested were unable to rescue this sort of excessive axon growth. So certainly 3 of these 4 variants were loss of function. It was left to us, I guess, to really see if we could then link our patients with the intellectual disability, mild intellectual disability, and check whether they had any evidence of the vitamin B12 deficiency. And when we used a highly controlled system where we expressed the variants at very high levels, up to 150-fold by mRNA, to see whether we could reveal some dominant negative effects, we can see that 2 of the variants here with the stars on them actually acted as a dominant negative effect and suppressed the expression of MACHC, although it was very mild. This other variant, which is the other one that had loss of function, is the nuclear localization variant. So being excluded from the nucleus sort of means that it perhaps doesn't act in a dominant negative manner in this particular assay. So there was a mild effect in this highly controlled system on MACHC. Again, we went back to the patient cell lines to really interrogate whether there was differences in MACHC expression in the patient cell lines. And what we found was that in these cases that we had that had basically mild intellectual disability, there was no evidence for decreased MACHC expression, with the exception of the nuclear localization variant, which sort of showed a slight depression. When we went back through the clinical records, we also obviously asked the question on whether there was any evidence of vitamin B12 deficiency, And the clinicians come back to us and said that essentially there was no evidence, except for, again, this nuclear localization variant where there was a transient bout of vitamin B12 deficiency that ended up fixing itself. So in a way, we were left with what we had. These variants, they had mild intellectual disability, and that were caused by loss of function mutations in HCFC1, but they had no evidence of the vitamin B12 deficiency. So we wondered whether these two features could somehow be dissociated. And we had kind of, in a way, a really nice assay because we'd knocked down modestly HCFC1 in neural progenitor cells and also in neurons. And when we knocked down just this modest amount of HCFC1, we certainly saw defects in the behavior of these cells. But did we see differences in MMACHC expression? Well, we were able to check, and the answer is no. So in a way, certainly, it looks like there are perhaps, in some instances where the loss of function mutation is not severe enough, you can have a neurological defect without the metabolic defect. And so I guess I've just summarized this here, looking at it's kind of like a genotype phenotype, that in every individual you'll have a range of HCFC function based on expression and mutation. The neurological features, there are two thresholds. When we have overexpression, as we saw in the non-coding variant, you get mild ID. And then when you have missense variants that are loss of function, you get a range from mild ID in seizures, which is the variant that we studied, right down to really severe ID and intractable epilepsy, which is the features that were also associated with the metabolic outcomes. So we think the threshold for the metabolic outcomes is much lower. It's only when you get really severe loss of function mutations in HCFC1 that you'll have the combination of severe ID and the cobalamin deficiency. So I just wanted to wrap up. Hopefully I've highlighted to you some of the ways which we're using information from patient-derived cells, typically used for gene discovery, but when you're looking at variants in genes which have an effect on the transcriptome, having a look at these cells can be really influential, guiding your subsequent studies in other cell culture models to really understand how these variants can affect the ability of the brain to grow in a semi-high throughput manner in the dish. Of course, now with this information, we're investing into more sophisticated models of induced pluripotent stem cells and small animal models, which will actually give us some nice physiological readouts. But I guess I wanted to just stop there. I hope you've enjoyed the two stories I mentioned to you today. The work's obviously done by a lot of people. I would quickly just mention the members of the neurogenetics laboratory, Lam Sam Nguyen, who did the majority of the RNA sequencing analysis, and the many collaborators from overseas. And of course, the work couldn't help without the support of the affected individuals and their families. So thank you very much.
Thanks very much, Dr. Jolly. We're bumping up against time, but we do have a couple questions already. And if anybody else would like to send their questions in, please do so. So Lachlan, one of the first questions I have is you've shown changes in both localization and regulation of UPF3B mRNA. What role do you think localization plays in the regulation of UPF3B?
Yeah, that's a really interesting question that I guess at this point in time it's just an observation, but it really requires functional interrogation. So it's something that we're interested in. I guess what would be an appropriate experiment to really get to the bottom of that would be a reporter gene that reports on the activity of NMD, and then to really work out whether the output of the NMD pathway is really affected by different variants of UPF3B, which you can force to be included or excluded from the nucleus. So at this point in time, we just don't have a functional readout to associate with that observation, but it's certainly something that's really intriguing and hadn't been observed before.
Perfect, thank you. You showed NMD activity is affected by CNVs, mutations in UPF3B as well as USP9X. Do you have any evidence that this pathway is regulated in an epigenetic fashion as well?
Indirectly, yes. So one of the things we are a little bit interested in is looking at which genes downstream of the NMD pathway that are really deregulated. And in the brain, there's some evidence coming out that there are a lot of genes, obviously there are a lot of genes in the brain which have alternative splicing. And this introduces a number of new NMD targets, so what's called alternative splicing associated NMD. So this is quite prevalent in the brain, and a lot of these target genes in the brain that undergo this alternative splicing that introduce NMD features, when you do gene ontology on that set of genes, you become really enriched with genes that affect the chromatin and a lot of methylases. So we think that there's most likely a link between NMD and the epigenetics. Obviously, with intellectual disability, which is the outcome of defective NMD in these patients, it affects their ability of learning and memory. So the association with the role of epigenetics in the learning and memory process is certainly already established. And I guess by looking at some of these particular NMD targets which have roles in epigenetic modification will be the way to really try and resolve that link.
Okay, thank you. And I got time for one more question. So you showed UPF3B function and the genes that it influences, right, and the role they play in central nervous system development, neural development. Any additional insight into those genes? Do they fall into similar pathways? Are they particular regulators? Do you have additional insight into the function of these genes?
In terms of, so I guess the question is, when we have the list of deregulated genes that we found in the affected individuals, could they group to a particular process? The answer for that is no. There are lots of I guess, I mean, they do group to things like brain development, but there's no particular pathway which really stands out. For example, you know, they're all downstream effectors of the Wnt signaling pathway, or they're all involved with synaptogenesis. So at the moment, there's no sort of way to boil down the sort of global information that we have to be really certain of a particular role of NMD in brain development. But there are certainly a lot of interesting data coming out on the role of NMD in particular processes of brain development, axon guidance, neural progenitor cell proliferation, which I saw even earlier stem cell differentiation into cells of the nervous system. So I think it really awaits a system, which we're trying to do now, is to really have a closer look at the transcriptome differences in cell types which are relevant to the disorder, that being embryonic stem cell derived neurons. So these are human cells, they're of neural nature, and I think that's a perfect platform to really interrogate which particular processes, in particular with reference to brain development, are affected.
Perfect. Okay. Well, thank you. So we are right at time, everybody. I appreciate you joining the call. We will have a recorded version of this, which we'll send by email, and you can also find on the Illumina website. So thanks again for your time, everybody, and have a great day.